Hello, the next portion, according to our syllabus, is amino acids. So let's start from it. Okay. Now, what is an amino acid? Amino acids, often referred to as the building blocks of proteins. Proteins are macromolecules, which constituent compounds are these amino acids. And these amino acids play many critical roles in our body. So there are more than 300 different amino acids in the nature, but just 20 amino acids are found as constituents of mammalian proteins. So what is protein? I told you, protein is a large biomolecules or macromolecules consisting of one or more long chains of amino acid residues. So proteins has many different functions in our body, such as catalyzing function, for example, transporting features, or also it's providing structure to cells, etc. Now I want you to distinguish the difference between the macromolecule and micromolecule. So in biology, micromolecule refers to a molecule of relatively small size and low molecular weight, uh, as opposed to a macromolecule, which is large and with greater molecular weight. So, in short, micromolecules are small molecules which are constituents of the macromolecule. For example, our amino acids are micromolecules which are participate to build these macromolecules such as protein. So, once again, macromolecules are molecules which contains a very large numbers of atoms, such as protein, and micromolecules, a molecules that is relatively smaller than macromolecule and have lower molecular weight. For example, glucose are the monomers of glycogen. Glucose is a micromolecule, whereas glycogen is a macromolecule, and also amino acids are micromolecules, and they are monomers of macromolecule protein. Okay, now let's start from the general formula of these amino acids. So, amino acids are organic compounds that contain amine group, also carboxyl group, and side chain. And this side chain known as an R group also. It's an alpha carbon, and I will talk about this alpha carbon. So under the physiological conditions on physiological pH, this amino group can be ionized. Also, this carboxyl group ionized, and it gives out COO minus and NH3 plus. So when we studied buffers, we talked about the buffering properties of proteins. So proteins consist amino acids, yes, many amino acids together. It's a sequence of amino acids. So this amino part was referred as an basic part of this protein, and this carboxyl group was known as an acidic part of this protein. So here you can see that at physiologic pH, and this physiological pH is 7.4, this amino group is dissociated, forming a negatively charged carboxylate ion here. It's known as an acidic part because it can donate the hydrogen, and amino and amino group, which is protonated, it was referred as basic part, and it can accept the hydrogen. So it's a side chain of this amino acid, and it's an alpha carbon. Now, what is the alpha carbon? It's the second carbon after this carboxyl end, which is attached to this carboxyl group, also to the amino group, and to the side chain. So here is a rule about this alpha carbon, and this alpha carbon is an atom next to the carboxyl group, which is therefore numbered the second in the carbon chain. Starting from that functional group is called the alpha carbon. Okay, now two amino acids are joined together with the peptide linkage. So in proteins, amino acids make a polypeptide chain. Yes, amino acid sequence can make a polypeptide chain of proteins. So this amino acid joined together with the peptide one. You can see here the amino acid 1. It's an amino acid 2. And via this carboxyl group, oxygen, and amino group 2 hydrogen, they can give out the one water molecule, and here the linkage is known, this linkage is known as a peptide bond. So in the peptide bond, you can see CO and H, and it's the N-terminal part of this dipeptide, and it's a C-terminal part of this dipeptide. Dipeptide means that in that case, just 2 amino acids are joined together. Tripeptide means when there are 3 amino acids together joined, and polypeptide means more than 10 amino acids joined together with this peptide bond. So with the reaction, it looks like that. It's a deprotonated part, COO minus, and it's a protonated part. So here, this 1 oxygen and 2 hydrogen from this amino part joined together gives out 1 molecule water, and this linkage given in red is known as a peptide bond. So here is the N-terminal beginning of the chain, and it's a C-terminal end of the chain. Okay, now let's talk about the side chain. Side chain is responsible for the properties of the amino acids. So all amino acids, just one exception is the proline, are identical. So just the side chain are different, or this side chain are known as an R group also. They are different in their charge, shape, polarity, and side, and gives each amino acid its unique biochemical properties. So amino acid side chains are typically grouped into non-polar, polar, acidic, or basic categories. And it's known as a classification of an amino acid according to their side chain. So there are given classification, and this classification are different, but in our book is given 4 different classes according to the side chain of this amino acid, and they are non-polar, polar, acidic, and basic. Now, we know what is acidic and we know what is basic, yes? But what's the difference between polar and non-polar amino acids? Non-polar, it means that they have an even distribution of electrons at the physiological pH. But polar, they have an uneven distribution of electrons, such as acids or bases, yes? So Here is given non-polar side chain amino acids. They are glycine, alanine, valine, leucine, isoleucine, phenylalanine, tryptophan, methionine, and proline. About proline we will talk because it's an exception among the amino acids. So non-polar. It means that they does not gain or lose protons, and they does not participate in hydrogen or ionic bond. So they are referred as lipid-like or oil-like or hydrophobic amino acids. You can see here is a glycine, is the smallest amino acid. It has the smallest side chain. It has just hydrogen. It's an alanine. Its side chain is CH3. It's a valine. And I want you to pay attention on this tryptophan. Tryptophan is an amino acid with a bulky side chain. Yes, it's a side chain. And this part is the same for all amino acids. Yes, you can see. And also pay attention on the methionine. It's a sulfur containing amino acid. Its side chain is given here. So amino acids with non-polar side chain. I told you they don't gain or lose protons or participate in hydrogen or ionic bonds. And they are thought as an oily or lipid-like, a property that promotes hydrophobic interaction. Now, what is the hydrophobic interaction? Hydrophobic interaction, these amino acids can make the hydrophobic interaction between each other. They can cluster together in the interior side of the protein. So this non-polar side chain fill up the interior of the folded protein and help give it to its three-dimensional shape as it's given here. So it's a soluble protein. In these yellow parts is a non-polar amino acids. They are hydrophobic, and you can see that they are clustered in the interior side of this protein. So these are polar amino acids. They are hydrophilic. Hydrophobia, it means that they are afraid to water, and hydrophilia, it means that they like. They can they are polar, they are water soluble. So pay attention that these hydrophilic or polar amino acids are clustered on the surface of this protein. So here it's given another type of protein, it's a membrane protein, and here you can see it's a lipid bilayer. So cell membrane contains a lipid bilayer. So the lipids has a hydrophilic head and hydrophobic tails inside. So in the hydrophobic tail among them, they are clustered together on the inside of this lipid bilayer where these hydrophobic tails are given. So they can give a hydrophobic interaction between each other. So now let's talk about rule. What is hydrophobic interaction? It describes the relation between water and hydrophobes, low water soluble molecules. So hydrophobes are non-polar molecules as our amino acids was given there. So and usually have a long chain of carbons that do not interact with water molecules. So the good example of mixing of fat or oil and water to obtain this interaction. Yes. Okay. Now let's talk about the simplest amino acid. It's a glycine. So this part is the same for all types of amino acids. Just one exception is proline, and it's a side chain of this glycine. Glycine has the simplest, smallest side chain as just one hydrogen atom. So is referred as the simplest amino acid. So it's our proline. It's referred as an amino acid rather than amino acid. Why? Because it has an amino group and other in compared to the alanine. Alanine has an amino group. It has an amino group. So this nitrogen also participates in this. It's a part of this five-membered ring, whereas the other amino acid has a free amino group here. So it's an exception, this proline, and so this proline participates to in the pro-polypeptide chain also, and they can make kings or turns, and we will talk about it while we refer the polypeptide chain and the proteins. So now we have a polar amino acids here. Polar amino acids, it means that they have zero net charge at physiological pH. So here we have serine, threonine, tyrosine, asparagine, glutamine, and cysteine. So serine, threonine, and tyrosine, they have a polar OH groups. So they can make a hydrogen bond with water or other molecules. So here we have also asparagine, glutamine, and they have amide group, which are now protonated. They are polar amino acids. So also these three amino acids can make a hydrogen bond. That's why they are referred as polar. So I want to talk about the cysteine. In the previous slide, when we talked about the non-polar amino acid, I talked about the methionine. Methionine, it also contains the sulfur, and the second sulfur-containing amino acid is the cysteine. And cysteine can make an disulfide bridges two cysteines in the polypeptide chain. It has a thiol group. It's a thiol group, and so two cysteines can make the disulfide interaction. But methionine contains just sulfur and CH3 methyl group, so methionine can't make disulfide interaction with other sulfur-containing cysteines. Just cysteine is participating in these bonds. So what is this disulfide bond? Two cysteines with its thiol group is presented here. Participate in the disulfide interaction. It's an oxidation of it, and it's a reduction. We know that loss of electrons is an oxidation and reduction is a gain of electrons. So here is a two cysteines which are, for example, part of this one first cysteine and the part of one polypeptide chain. The second cysteine is a part of the second polypeptide chain, and they can stabilize these two polypeptides by this disulfide interaction, or it's known as a disulfide bridges. Many extracellular proteins are stabilized by disulfide bonds. For example, it's an albumin, it's a blood protein, and albumin has a many function. It can transport different insoluble, for example, fats in the blood. So here we have amino acids with acidic side chain. Yes, it's an aspartic acid and glutamic acid. So they are both proton donors. So that's why they are acidic. Yes. So, for example, at the physiological pH, this aspartic acid can donate its hydrogen, also this glutamic acid, and they are ionized at COO minus, both of them. So here we have the second part of this classification, they are basic amino acids. So these basic amino acids are histidine, lysine, and arginine. And pay attention, please. Here they have an imino group, amide group, and they are protonated. They are charged plus. It means that they can accept hydrogen. So they here we have a histidine also. Histidine is a weakly basic at physiological pH, but at the polypeptide chain it has either positive or either neutral charge. Okay, now let's talk about the names of the amino acids, abbreviations and symbols. So each amino acid name has an associated three-letter abbreviation and also one-letter symbol. So if only one amino acid begins with the particular letter, then that letter is used as a symbol. For example, it's known as a unique first letter, yes? Cysteine. Cysteine is a three-letter abbreviation is CYS, cis. But the one-letter unique first letter is C. Histidine, H. Isoleucine, three-letter abbreviation is goes I and LE, and the one-letter unique first letter is given as I. Yes, I means isoleucine. Methionine, M, serine, S, and valine, V. But most commonly occurring amino acids have priority. So if more than one amino acid begins with a particular letter, the most common of these amino acids receives this letter as a symbol. For example, alanine and alanine. Wait, please wait. Okay, alanine and asparagine. Yes. Alanine is the most common amino acid given, so this A belongs to the alanine. Also, there are two amino acids such as glycine and glutamate, but glycine is more common, so this G belongs to the glycine. Also here we have leucine and the letter is L, proline, threonine, etc. Okay. Here we have a similar sounding names. So it may be sometimes confusing. I will try to explain how to remember these names. Arginine, it starts with R. So it's the first unique letter is R. Asparagine, so it sounds as an N, yes? Aspartate. So it's aspartate, I told you, it's an aspartic acid. Yes, aspartic is like known as a D. So here is a glutamate. So it's referred as E. Glutamine, it sometimes sounds as Q. Yes, glutamine. So its unique first letter is Q. Phenylalanine, it's F. Tyrosine, it's Y due to the second letter, and tryptophan. And pay attention, it's double, YW. I told you that tryptophan is an amino acid with bulky side chain, and its side chain is a double ring in the molecule. So now about the aspartate or asparagine. It's B near to A, so it's known as a B. Glutamate, it's known as an E, that because of this X, it's a three-letter abbreviation. Lysine is known as an K, it's near the L, and Undetermined amino acid is known as X. So this X can be equal to glycine, etc. For example, we have while we study the collagen, this is given, for example, glycine and X. So this X can be equal to the hydroxyproline or hydroxylysine. So this X is an undetermined amino acid. So now let's talk about the optical properties of amino acids. There are two different types of amino acids. They are D form and L form. And these D and L form, it's an arginine, alanine given here, are different in they are stereoisomers. They are mirror images of each other. They are also known as an enantiomers. So they are optical isomers. What is isomerism? Isomerization when they are the same formula, but these D and L alanine are different in their properties. So D and L alanine are the mirror images of each other. So in the body, in the mammalian proteins, just L amino acids are presented. So here is given that amino acids, they have an asymmetric center at the alpha carbon, can exist in two forms, L and D form. So the two forms in each pair are termed stereoisomers, optical isomers, or enantiomers. So we talked about the isomers. Now I want to show a polypeptide chain. So polypeptide chain is a many amino acids joined together with the peptide bond. So it's macromolecule, and this one amino acid is a micromolecule. So each polypeptide chain is a series of amino acids joined together in a specific sequence. So each amino acid has its unique R group. A particular amino acid sequence creates a polypeptide whose function is dependent upon its chemical structure. It's a polypeptide, it's an alpha helix here, and we will talk about the structure of proteins. Now I want to say what is amino acid residue. So it's an our a polypeptide chain. This polypeptide chain has a head. It's known as an N-terminal now because in this side we have an amino group of this amino acid, and the last tail is known as a C-terminal because here is presented COOH or COO when it's ionized group. Yes. So the linking of two amino acids is accompanied by the loss of one molecule of water. Yes, if you remember from the reaction. Yes. A series of amino acids joined by the peptide bond form a polypeptide chain. So in this polypeptide chain, each amino acid unit is known as an amino acid residue. Why it's residue? Because this amino acid, for example, lost its oxygen, and this amino acid lost its two hydrogen ion, and one water molecule is released. So it's they are after that they are referred as an amino acid residue. Now a peptide is two or more amino acids joined together by peptide bonds. It's a summary. A polypeptide is a chain of many amino acids which are joined with the peptide bonds. Yes, protein contains one or more polypeptides. Therefore, proteins are long chain of amino acids held together by the peptide bonds. So here is a polypeptide chain with different amino acids, and he is given ionic bond. Ionic bond between two amino acid side chain. So this side chain is a side chain of one amino acid, it's a side chain of another amino acid, and they can bind with an ionic bond. It's an interaction between the positively charged and negatively charged ions. So they can interact each other, yes? Here is the two cysteines are participating in this bonding. It's a disulfide bridge, and here is a sulfur-containing cysteines, and they can interact and make a disulfide bridges. So here we have a hydrophobic interaction. Hydrophobic interaction, it means that the amino acids are non-polar amino acids participating in this interaction, and they are clustered in the interior side of the protein. And we have also a hydrogen bond. And we know what is hydrogen bond. Hydrogen bond is a bond between the oxygen or hydrogen and nitrogen, etc. So here in general, it's given the interactions in this polypeptide chain via the amino acid side chains. So it's not given in your book, but I added this information in the presentation. There are three different groups beside of this classification of amino acids. They are essential amino acids, non-essential amino acids, and conditional amino acids. Now, what's the difference now between them? Let's start from the essential amino acids. They are amino acids which can be made by the body, and we should obtain them from the diet, from the food, yes? So there are 9 essential amino acids. They are histidine, leucine, lysine, methionine, phenylalanine, tryptophan, valine, etc. So now about non-essential amino acids. Non-essential amino acids are those which we can produce in our body. So we don't need to get them from food. Yes, our body can produce these amino acids. So non-essential amino acids are alanine, arginine, asparagine, aspartic acid, cysteine, glutamine, glycine, glutamic acid, choline, serine, and tyrosine. But here is a third type. It's a conditional amino acids. It means that they are usually not essential, but if there are sometimes happen due to illness or stress, our body can't produce them. So in that case, we need to obtain them from food. So their synthesis can be limited under special pathological conditions. So these conditional amino acids are arginine, cysteine, glutamine, tyrosine, glycine, ornithine. It's not given in the classification, but it's referred as an essential conditional amino acid here, also proline and serine. So it was about everything about the amino acid, what was given in your book. So you should prepare this material from the Leaping Code Illustrated Reviewers. Here is given the pages which you need to prepare for the Saturday tutorial. Please collect your questions and let's make a discussion more interesting. So you can find this book on my shareable link of my drive, and please prepare it. Thank you.